Good morning, everyone. Welcome to this webcast. We're glad to have you join us this morning for CSU course-based undergraduate research experiences, CURES. I'm your host, Jennifer Wicks, coming to you live from Central Station U Studio here at the Office of the Chancellor. We're looking forward to your comments and questions in the chat pod over to the left of your screen throughout today's session. And thank you for putting in your answers to the polls on the screen. Number one, which campus are you joining from today? As you can see, we have people joining from almost every campus across the system. And poll number two, are you planning to apply for a grant? We want to take a moment before we get started just to touch base with our footprints data savings. So we're going to switch over to that right now, and I'll ask you to type in your zip code from where you're joining. I'll send a note to your computer here. You'll see a pop-up that shows up on your screen here. If you could just type in that zip code from where you're joining us, and then dial in the number of people in your group. If you are in a group today, we have 4 of us here in the studio, actually 3 of us here, and we'll click on that there, and then save and close, and that'll bring us back to the main savings here. And so far we're saving 18,900 pounds of CO2, over 20,000 US dollars, and this is whether you would fly or drive to us here at the Office of the Chancellor for this session. And we're saving over 36,000 round-trip miles by using this webcast platform for today's session. Well, I'd like to introduce our facilitator today, our moderator in studio is Dr. Frank A. Gomez, the Executive Director for STEMnet here at the Office of the Chancellor. And with that, I will hand it over to Frank to begin. Frank.
Good morning, and welcome to another webcast. I'm Frank Gomez, Executive Director, and your host today of STEMnet. As our tech-centered and global economy continues to grow, and our need for a diverse STEM-educated workforce grows with it, there are strains in the mentored research model. Providing traditional research experiences for all students is impractical, if not impossible, at many institutions. So how can we furnish students with the opportunity to learn how to test a hypothesis and fail in a comfortable and structured environment? How can we create the next generation and beyond of leaders in STEM who appreciate the wonders of science, exploration, and who will solve tomorrow's biggest challenges? A key development has been the model of including an undergraduate research experience as part of the curriculum, and the course-based undergraduate research experience, CURE model, is one pedagogy that provides a research experience to more students than ever before. Infusing research in the curriculum is an opportunity with benefits to students, faculty, and institutions. Not only can we include and serve more students, but we can expand the passion for science that so many of us older students develop while practicing science. The secret sauce for the success of CURES and related practices is that they offer the same benefits to students that a mentored research experience does. The added advantages of the practice are that more students participate, they can participate much earlier in their careers, they can use multiple CURES experiences to shop for their passions within their discipline, and multiple CURES can be scaffolded over the course of a student's undergraduate program, reaping even more gains. All this while we know that those students are realizing the same benefits in their development, not to mention the possibility for more scientific discoveries. I'm excited today as we bring together CURE exemplars from our own CSU community to discuss their projects and from a myriad of disciplines. Before we begin, I want to remind the audience to use the open chat box on their screens to type in their questions. I'll collect the questions in addition to the questions shared during the registration process for this webcast and moderate the Q&A period. May I add, questions we are unable to address during the webcast or submitted during the registration process will be answered and made available to registrants in a few weeks. Let's begin the presentations with Amelia Vankeuren, Assistant Professor of Geology from Sac State. Amelia, the floor is yours.
Thank you. I'm excited to be here today to talk about how we at Sacramento State are getting serious about geology by using CURES investigating human impacts on the local environment. Shown here are images from courses that I teach in which students are collecting water samples from local rivers and monitoring groundwater elevation in wells on campus to see how we are affecting the local water supply and water quality. This is part of a larger effort. SIRIUS stands for the Sustainable Interdisciplinary Research to Inspire Undergraduate Success, It's a program that started in biology, but has expanded into chemistry, environmental studies, and geology. So it's really a campus-wide effort to put cures in STEM classes. This effort is supported in part by NSF multiple grants, including a major research instrumentation one, the W.M. Keck Foundation, and CSUPERB. I'll be focusing today on the geology classes. These include physical geology, a lower division general education course, in which students are mostly non-science majors, and this gives them an opportunity to have an authentic science experience. And then two courses that I teach, hydrogeology and environmental field methods, which are upper division geology electives, and the environmental field methods course is taught in collaboration with environmental studies. The first course, environmental field methods, is designed for students to get hands-on experience with equipment and techniques that they will use in their careers in environmental geology. This could include environmental consulting or working for local, state, or federal agencies. The course topics include sampling and characterization of soil, surface and groundwater, as well as air quality. They get experience with sampling design and quality control and quality assurance metrics. It's a relatively small class with about 15 students and is a mixture of both upper division undergraduates as well as some graduate students. In this class, the students get experience through cures embedded in a 3-hour weekly lab section. This includes 2 to 4 labs in which the students collect water samples from local rivers to look at nutrients and pesticides and see the spatial differences in these two. The samples are collected by both geology and environmental studies students and are then transferred to chemistry students for analysis in their quantitative analysis course. So that equipment was funded by the NSF Major Research Instrumentation Grant and the Keck Foundation Grant. And so it's really a combination of 3 different classes all working together to look at water quality in these local rivers. I've also had students contribute to my own research through sample collection. I am monitoring a project in which we're doing managed aquifer recharge, and one question is how water quality is affected by putting extra water in the groundwater system. So I brought students to my local sites and they collected samples using industry standard protocol that they had learned in class. The students seemed to really enjoy the fact that the samples they collected were going to be directly incorporated into research projects and reports for local agencies. They also gained valuable experience with the real equipment that they will use in their jobs. In the next slide, I will discuss how we have a final cure in which students lead their own project evaluating air quality on campus. So an example of this is, do construction projects on campus significantly increase fine particulate matter, and are those levels hazardous to human health? In this, students work in small groups and they follow the project really from start to finish. So they develop their own research question, do hypothesis development, they implement sampling design, they do data collection with handheld meters, and they ensure that they're following and meeting their quality assurance and quality control metrics. They then do data interpretation, and finally write up their reports in the form of a scientific paper, and this is written iteratively. So each week they submit a section, I give them feedback, and then they also do peer review on these reports before submitting a final revised copy. The next course is hydrogeology. In this class, the focus is really on groundwater, and so we look at groundwater flow, contaminant transport and remediation, and water quality. It's a slightly larger class, up to 25 students, and is primarily upper division undergraduates in the geology department. We're very fortunate at Sacramento State to be having one of the largest on-campus well fields in the nation. We have 19 monitoring wells and an extraction well, purely for educational purposes. So shown here are students who are measuring the water level elevation and doing aquifer testing within these wells. So these students are again getting hands-on experience with equipment and also software. These cures are embedded in the weekly lab section, and we again start with mini cures. So doing aquifer testing to measure aquifer properties like permeability and storativity, where they're using software that they might use when they get to their next job. And then they also collect groundwater samples and do some preliminary chemical analysis in order to evaluate if there are changes in groundwater quality over the course of time. And we finally culminate in a whole class research project. For this, instead of working in small groups on separate projects, the students vote to address one larger question as a class, and then break into smaller groups to work on separate aspects of that question. So an example of a larger question is, does the water flow and water quality in the Lower American River affect groundwater on campus? Then a small group might address changes in water quality over time. We're fortunate our wells were put in 1990, and we have a sparse but long 30-year data set in water quality. And we also have pressure transducers to monitor changes in water level elevation and local data for river stage over time. So different groups might focus on one of these aspects. One benefit of this is that students get to leverage their existing strengths. So if there are students who already have GIS experience, they can use that. Students with coding experience in R can use those techniques to manage large data sets and to pull out important pieces of information. And we use data generated both through previous student research projects and prior years, and also local publicly available data from state agencies, local agencies, and the U.S. Geological Survey Water Science Center, which is conveniently located in our building. So some results from implementing these cures: I've noticed a difference, an improvement in courses. Students do see themselves as doing real science. So we have assessment data, which was helpfully put together through the SIRIUS project. So there were already existing questionnaires to assess whether or not students felt like they were doing authentic research that we were able to incorporate in my class. So in the pre-cure questionnaire, about 75% of students felt they were doing real science. And after the cure, it was up to 93%. And I should note that the 75% was already probably a relatively high number because prior to a cure, the course was a project-based service learning using U.S. Geological Survey data. I've noticed that because students are working on a common research goal, there's a more collaborative class environment, and we see increased interaction between undergraduate and graduate students, which enables the undergrads to talk to the graduate students and find out what graduate school is like, and hopefully generate interest in graduate school for themselves. And I have noticed better class engagement. Students see that the work is directly applicable, and they are more interested in it. So lessons learned: I've found that cures require flexibility. The data certainly aren't always what you expect, and the students do get frustrated when data collection or analysis doesn't go smoothly. So you really have to prepare them for that contingency. The cures are time intensive, so I did have to cut some content to enable us to fit in these authentic research experiences. It's really valuable to start assessment before you start implementing the cure so that you have a before and after comparison. And again, I really benefited from that collaboration with other departments who had already done cures and had some of those materials developed. And to implement this incrementally. So you don't have to have your cure completely worked out before you start. In hydrogeology, the first time I taught it, I just got the labs together where they were doing hands-on experience with real equipment and real samples. And then the next time I taught it, I added on this overarching research question. So I strongly encourage collaboration and really have fun with it. The next steps are going to be to continue to record data for the local environment and develop a long-term data set on groundwater quality and elevation and air quality. We'll continue to include these cures and adapt them as necessary and support the equipment necessary through small course fees. And I hope to eventually have enough data to support the addressing larger research questions and even producing manuscripts with undergraduate co-authors. So in summary, these cures in geology were quite successful, and we evaluate the impact of the campus population on the local environment, including air quality, and water supply. The students feel like they're doing real science as well as getting hands-on experience, and the interesting research questions help keep both the students and me as the instructor engaged. All right, thank you for your attention. I'd be happy to answer questions at the end of the webcast. And now I'd like to introduce David Rhoads from California State University, San Bernardino.
Hey everybody, I'm going to talk to you today about our cure that we developed on functional microbial genomics. And this is a collaboration between myself, Steve Slater, Brad Goodner, and Derek Wood. And this was initiated as part of an NSF grant to develop cure projects. So while I'm going to go over some of the details about the cure, one of the things I want to emphasize is the overall approach, that is very flexible and expandable. So the overall research approach, some of the details of that, we're trying to verify predictions or annotations for genes encoding enzymes in amino acid biosynthetic pathways in the genomes of several agrobacterium strains. So the overall approach, we identify a biosynthetic pathway and genes for study. Then we obtain the genomic sequences for these from the genome databases for the genes. And then we clone the genes and test for complementation. So this is done as part of about an 8-hour-a-week lab class. And generally, we have probably about 10 to maybe 20 students in the class, offered once a year here at CSUSB. Okay, so some of the details for one particular example of a project, we're looking at cloning and testing genes for the P5CR enzyme in the proline biosynthetic pathway. So first, the students go to the database and search for potential genes, and I'll give you more details on that in a second. Then cloning the genes by designing primers based on the genomic sequences that they obtain. Then PCR amplification, and cloning into expression vectors. So then those genes are transformed into a construct, the construct is transformed into a mutant E. coli that lack the Pro-C gene. So then they can be selected for the ability to synthesize proline. That is, if they grow in the absence of added proline. So some of the little details of the project. This is one example of the proline biosynthetic pathway. So proline is synthesized from alpha-ketoglutarate. So we chose these based on a couple of criteria. One, that the enzymes have several potential genes in the agrobacterium strains that we're looking at. Secondly, that we have a mutant available in E. coli that we can use for the functional complementation testing. So we have to satisfy those criteria first. And usually the students are given some options for the different pathways before we actually start it. Then we use the amino acid sequence from the E. coli gene, and the students search through the databases to try to identify potential genes in agrobacterium. And then the genomic sequences are identified so that primers can be designed to amplify and clone the genes. And here's just an example of some of the data. So generally what we do is, if it's a small enough class, each of the students individually gets a gene to work on. But if it's a little bit larger class, then maybe we'll have a pair of students. At most, we try to work on one given gene. And this shows the data, successful cloning of the genes, of 9 different genes from agrobacterium strains that potentially encode the P5CR enzyme. So then the next step is to transform those into a mutant E. coli line. And so this is just showing at the top the plates where the cells are growing on plates that have proline present. And then the bottom row is plating the cells on plates that lack proline, and only the cells that can produce the proline from the cloned gene actually complement the mutation are able to grow on those plates. And of course, we have the requisite positive and negative controls of the mutant, and the mutant transformed with the E. coli gene. So the conclusions from this particular experiment, so all 9 of the Pro-C genes that we identified from the database were successfully cloned and tested for complementation, and 5 of the 9 genes were able to complement the mutation, while 4 of them were not. And part of the interesting part for the students is that one of the genes that was highly predicted to have a high similarity actually did not complement, which was kind of a surprise, but good for them to see in a real-world type of experiment. So then this is in preparation for resubmission for publication. So some of the bigger lessons learned by running this, and so we've been doing this for about overall approach for about 10 years now. So students benefit overall from participating in the authentic research cure in general. But more importantly, I would say that some of these, this type of approach can be used as a stepping stone for students to become involved in research. And often we find that students that are attracted to this class are ones that are maybe less likely, maybe don't have the capability to participate in an apprentice type research where they work more one-on-one with the faculty members in the lab. But this gives them a chance to participate in a real authentic type research program. Of course, the main idea of the cures. From our assessments, we found that participation in these authentic undergraduate cures has a moderate but positive influence on scientific inquiry competency, and also participation in authentic research courses did not predict scientific literacy skills, but did predict student interest and student attitudes. So part of the problem that we found partway through this cure was that we actually needed a better assessment tool for content knowledge. And what we've since discovered is the biomaps tool, and we're trying to incorporate that right now. Also, some of the project and course design and implementation are key components to setting up the cure, so designing the overall experiments and designing the curriculum. And finally, assessment must be designed early on in the process. So some of our next steps, so this is, I'm getting at part of the advantage to this kind of approach is the robustness of the approach. So there are hundreds of genes encoding enzymes for amino acid biosynthesis in bacteria, not all of which meet our criteria, but many of them do. And so examining each one of these is a project in itself. And what we found that it usually takes about 3 or 4 iterations to complete. And also, I generally supplement the research with actual apprentice type research in my lab just to keep things moving along a little bit faster. So while the specific genes can be changed, the overall approach is the same, and so it takes minimal effort to develop new projects and continue on. So this is just examples of some of the different pathways that we're working on. So the one pathway has already been completed in pink here, while the green one we've already started, and then the blue ones are ones we're planning to do in the future. So you can see the idea of the robustness of the overall project, or this overall approach. So in summary, if designed properly, cure projects are feasible approaches to obtain original research data. Students benefit from participating in the cures involved in authentic research. Cures can be an excellent stepping stone for students who might not otherwise get involved in an apprentice type to become involved in authentic research. And finally, assessment must be designed early in the process, and effective assessment tools are critical. Okay, so with that, I'd like to introduce our next speaker, Kim Coble from San Francisco State University. Kim.
Hi, thanks everybody for having me here. I want to give a quick shout out to my collaborators from a bunch of different institutions, but especially Lynn Kaminsky and her group at Sonoma State, because I know they're in the audience here. Just a little bit about myself. This is my 4th year at San Francisco State. I came here from the Illinois State system, where I was at Chicago State for 12 years. Early in my career, I was an observational cosmologist, and then I switched to physics and astronomy education research. So I'm going to tell you about several astronomy projects. Some of them are done with lower division introductory astronomy general education students, and some of them are done with upper division astronomy majors. All of these projects are spread over typically half a semester in either lecture courses or lab courses or courses that are lecture and lab combined. So the Global Telescope Network is part of Skynet, and there are a number of small-ish telescopes on the network. And we wanted students in the introductory general education courses to experience realistic practices used by real astronomers. So we had the students learn how to use software to determine what objects would be visible in the sky and when. We did that through a lab where they learned how to use the software, and then they created observing proposals to use the telescopes. Now, these proposals were scaled down like 3 pages rather than like a full-on observing proposal that a scientist would do. The students also then peer-reviewed each other's work, both in written reviews and then also a panel discussion based on NSF criteria. And you'll see many students mention that panel discussion as one of the most fun parts of the project. Then they collected their data. The telescopes are all operated online. And then with a minimum of data analysis, they presented their results to the class. One of my master's students at San Francisco State analyzed students' reflection essays. At the end of the process, they did a 1-page of things they liked, didn't like, or would change about the project. Each one of these categories has a breakdown. So many students mentioned positive outcomes. And there was a bunch of stuff about the process of science, being a real scientist. So these are some quotes to the side just to sort of illustrate. We also did interviews with students, and many students said it was extremely valuable to get their hands on real data. Also, quite a number of students had positive affect, not just interest, but also self-efficacy. There were a handful of things students didn't like, mostly having to do with real-world difficulties, like I couldn't observe my object because it wasn't up in the sky at this time of year. Another project we've done with general education intro astronomy students is called RIBSY. The URL for all the projects is there. This is using archival data from Kitt Peak and other places. Travis Rector at the University of Alaska and others have been working on this. And this is implemented at several different types of institutions in several different types of ways. So, you know, there's the full-on cure method, and then we also coined a phrase, Q-cures for quasi-cures, where it is still very interactive, but you don't necessarily publish the results. So these are readily available. They're especially good for astronomers, you know, who want to work with students who have a variety of interests, but maybe are not part of a large department. So our research questions here is how the students' perceptions of their confidence changed before and after, and how they felt the RIBSY curriculum was different than their previous experiences of science. And we had several measures for this. And, you know, we had findings aligned with the cures model, especially the increase in self-efficacy, the drawing their own conclusions, and the importance of community. In addition, we found the students said that this was a very meaningful and unique experience in contrast with their prior science experiences. And then finally, I teach an upper division cosmology class for majors. This is a writing class. So the class is designed to engage students in scientific writing. Astronomy has a huge amount of publicly available data. And so we have students interact with curated data and models, and then we set them loose in the databases to create their own project. And again, they go through the stages of a proposal, a peer review with the NSF style panel, and then they do their data analysis, or if they're creating a computational model, they do that. And then present their results and reflect on it. So again, we're seeing similar themes in the reflection essays, mentioning that it was, you know, much deeper and more realistic than doing lecture and problems and homework. And they enjoyed the struggle. They really enjoyed the real world aspect of it. They loved the NSF panel. You see some really high level thinking here with these students. So I am still analyzing these data, but I feel like this was a success. These students then go on to do REUs and other more intensive practices typically. So lessons learned. Astronomy is really conducive to having open source data and telescopes. It was super important for students to actually get real data because astronomy can feel like disconnected and too far out there. They really enjoyed the experience of peer review. This increased their self-efficacy and the believability of results. So our next steps are just to make the implementation easier for instructors who want to take up these projects and continue the research on the upper division projects similar to what we did for the lower division projects. Okay, so next up are Vadim Kaiser and Christopher Meyer.
Hello. Well, really excited to participate in this webinar, and I've learned a lot from the other presenters. And in fact, I think the CSU system is well poised to contribute to the national conversation on cures, and we have a lot to offer, clearly. So my colleague Vadim and I have a two-part presentation for you, which sort of includes an overview of the Developing Cures curriculum at Fresno State. It has a structure-function approach that I can't help as a recovering biochemist in the role of dean. You can see from our diverse college origins that the long-term intention is to engage students across disciplines in team science cures. I'll be introducing some of these concepts at sort of a 30,000-foot level. Vadim will bring them down to earth. He's going to share some compelling and provocative ideas and examples that he's developing beyond traditional lab experiences. And I'll briefly highlight diverse cures in our college, from the more familiar areas of biology and biochem to recent efforts in physics and computer science. And math and psychology are becoming part of that endeavor as well. It really takes a village, so I haven't mentioned all the collaborative strands here. A special shout out to Joe Ross, I think he's in the audience, who's done some really seminal work on genetics, molecular biology cure development. So briefly, Tricia Van Laar will be talking about some of her work, Teresa Brooks in biochemistry and chemistry, Pei Chung Ho in physics, and Mateen Peruz in computer science. And I'm proud to include Erin Dolan as a collaborator. She's, of course, the national expert on cures, founder of CureNet, and convened a NSF-funded Cures Institute on campus last summer that brought together people from all departments in the college. So I'm going to go kind of quickly through this. Some of this was well covered by Frank, and I think that I may be preaching to the converted here, but of course we have a very ambitious goal for every major to have a cures experience, both at the freshman, sophomore level and then in upper division. And as a more of a capstone experience, as we've heard from others. So we did initiate it in our college, and we wanted to transform some of our courses into scalable cures to really democratize that experience. And of course, putting it into upper division labs is a bit easier. That's low-hanging fruit because many people already do that. They may not call it cures, and they may not use all the cure steps. Lower division, I think, to be very impactful because they're the most vulnerable to dropping out of STEM majors, and often students might lack the social capital and the background to know the unwritten rules of joining a research lab. Maybe they work part-time. So embedding into the curriculum, as Frank said, outstanding way to broaden participation. There's a good buy-in for faculty, I think, to bring live research in the classroom, to collect data for proposals, presentations, publications, including students. It doesn't compete with their ideal, with their individual research labs, but rather enhances that by best preparing students. So we believe and Vadim and I are proof that the curriculum is transferable to other disciplines that can serve as a platform to really engage students in interdisciplinary and transdisciplinary research. So on our campus, that's risen to the level the comprehensive campaign, you always want to be included as a priority in that, right? For STEAM, and so STEAM, you could say agriculture, but we also mean arts and humanities, and we're very inclusive. So, and in fact, our comprehensive campaign reads that part of that will be include components for faculty fellows, postdocs. That's something that would really great for the CSU system, near-peer mentors, and it's also a vehicle to really, you have to have enhanced lab spaces in order, and innovative spaces in order to carry out cures. So I'll move a little quicker here. I'm going to leave plenty of time for my colleague Vadim. Just briefly, a slide I used to use when I was at NSF. Just what do we mean by defining disciplinary and interdisciplinary? So clearly a lot has been gained by disciplinary advances in these silos, and then multidisciplinary, which is defined as researchers working in parallel or sequentially from disciplinary specific areas. Well, as more tools and more data has become available, research working jointly from a disciplinary specific basis defines interdisciplinarity, and transdisciplinary, of course, the ultimate step of joint work using a shared conceptual framework, drawing approaches from the parent disciplines, and even creating new frameworks that transcend discipline boundaries. So that's what we'd like to see. And again, using bio through the lens of biology as an example, how do you get, for complex problems in the real world, how do you get people to understand the interfaces really between scales, from the molecular level, organismal, to ecosystem, the world, universe, etc. And I think it's good that we've had like microbial, functional genomics all the way to astronomy here. So how do we do that? And we'd like to establish as a distinction of our graduating students an experience in interdisciplinary team science and thinking across scales. So, and of course that needs to include other disciplines beyond STEM, and Vadim's going to be talking a lot more about that. So really briefly, our activities in the College of Science and Math included outward looking, where we brought in experts like Aaron Dolan, and also Brad Goodner, just to relate that to the San Bernardino work, who's a great collaborator as well. And there's some nice pictures of that. And cures in biology, of course, makes a lot of sense given the number of majors, as you've heard before. All the faculty they'd have to have about 60 students per lab, and that's not scalable or sustainable. And again, we have many part-time students. Some part-time students as well wouldn't have time for their work efforts. So, and we have a very diverse group that includes lots of first-generation students. So the idea here was to, I'll be speaking of the Bio 1A work that Trisha's working on with support from a Superb grant. And you can see that there are achievement gaps between first-gen students, Pell recipients, underrepresented minorities. Actually, women are doing better than men, you know. And so the key to that is transform the Bio 1A lab, into work with the model organism Tetrahymena, which is a great model system. And so the research student projects paired with the lecture will be using that area, which of course is well known for producing even Nobel Prize winning research in his active area of research. The students will choose roles as primary investigator, protocol expert, data expert, and analysis expert, and with groups of 5 students, and those roles and responsibilities will rotate. And I won't get into more detail about that, but I think it's very exciting. Computer science involved 3 courses so far, and bottom line on that's involved 51 students. Again, we have lots of computer science majors that can't be in individual labs, and impressively 4 international conferences and presentations. So I think that's going quite well. In physics, don't see as many cures in physics, but I'm really glad that this has been embraced. And you can see in a sophomore level, early junior level modern physics class, you can see all the different projects that are involved there, often with input from labs. So that's a great thing. part of the CURES landscape as reflected by others. You don't have to go all in all the way. You can start with a module, and ultimately, if you have a program, you would link and scaffolded courses, as you heard from Sacramento State. There's great opportunities for teaching in a CURE model that involves grad students, near-peer mentors, undergrads that have been through the class, as well as graduate students. So here published in JChem Ed, I won't go on and on about it, but it's a great opportunity to interface a trainee, which again could be a postdoc or grad student, in really live development. And what a great preparation to be a future faculty member. Would you like to say, well, I taught a class and I gave grades and it was Chem 1A or whatever, versus I developed a cure, here's where we presented the data, and a publication is in progress. So I'll just end with showing you what some questions are for faculty when in this topic of cures. Happy to follow up with anyone anytime after the webinar, and very pleased to introduce Vadim Kaiser.
Thank you so much, Chris. So I'm Dr. Vadim Kaiser. I work at the intersection between philosophy, science, and also art. And one of the interesting things is there are limited discipline connections between philosophy and the sciences. And in my research, I do interdisciplinary work. I try to model contradictory and disagreeing measurements. And a lot of what I do has to do with visualizing and modeling. So what I'm actually doing is trying to resolve unknowns and uncertainties. And so when I think in the classroom, how can we use visualization for students to be able to model work? One of the things that I do, and I don't know, Jennifer, could you load the small video, is so I go through a process of visualizing, and here we can just click on this. This is turtle measurements and disagreements in the field versus the laboratory versus computer simulations, and it's a lot of fun. So the next step is kind of, and I don't know if we could move or minimize this. Thank you so much. The next step is, well, how can students actually make their own models? How can they focus on causal diagrams, scale representations, but not only just the explanations of the real system, but also the methods behind those systems. So I really place a strong focus on robustness in methodology. And so the first step is, okay, well, we have to group students together. We have no laboratory, we have no funding, and we have to have these teams of scientists that are working on theoretical models. And the necessary conditions that I set, independence and perspective, unbiased communication, transparency about competence levels, and also, this is a very important one, you can only do this up to a certain number of students. So it's about teams of 4 or 5. And I'm just going to give you an abstract schematic of how one of these projects works. And the example that I'm going to use is memory in my cognitive science class. So first we start as a giant classroom, like one gigantic collaborative team, pull and parameterize data. We take a look at the relevant research. We try to pick out key variables and relations. And then we assign students, teams of 4 or 5, to model each scale. And this is really important because the students go back and forth with me iteratively in order to accurately model these scales. And so not only are students constructing these visual models, but they're also constructing these massive analytical documents where they focus not only on the explanations, but also on the methods behind those explanations. Now, once those scale models are completed, we have them side by side. And this is where it gets fun. We come together as a classroom again, and we look at the relations between scales. And this is really where a lot of unique research work emerges. So what students are doing is they're trying to synthesize complicated information in order to find causal conclusions between scales. And then each team of students has to recreate this process and submit it in the form of a visual model and also an analytical document. And the reason why this is so important is because, you know, this is something that I remember when you're taking a look at textbooks and you see models like the following. So this is a rough model of memory, and it's highly inaccurate, it's inadequate, it doesn't show you the methodological work. And I want students to really go through this total process of modeling so that they can actually be ready to do science. And so this is, an idealized version of what we do. We have pooled data, we turn it into scales and relations. And then we try to not only model the system itself, but also the methodology behind the system. And the two things that we're focusing on is modeling relations between scales, but also methodological modeling. So students are actually figuring out what it means for a methodology to be robust. And here's the final thing. Sometimes we construct just kind of a fun animation. Jennifer, could you please load the video? Yeah, and so this right here is just kind of a silly animation that shows you, so this is the physiological scale, and then you kind of zoom into here, you have the molecular scale, you look at activation, you look at molecular mechanisms, and you look at a process of memory deletion. You can actually delete memory, which is pretty cool, unless you introduce a novel event. So I think here we have a Sasquatch. And what ends up happening is you can reinforce the memory. And finally, we talk about that memory is determined by context. It's kind of an existential thought where context consumes memory. So it's not only scientifically, I think, important, but it's also kind of a little bit of fun. And so what we try to do overall is, with no resources, with no laboratory, we try to really expose students to models and methods so that they can work on complicated theoretical scientific work. Thank you very much. And so next I will introduce, oh, sorry, I moved that, Corin Slown and Corinne White.
Hi everyone, I'm really excited to be here to talk to you about the CURE scaffolding experience that we've been working on at California State University, Monterey Bay. My name is Corinne White. I'm the Curriculum Associate at California State University, Monterey Bay in the Undergraduate Research Opportunity Center. And I'm joined today by Dr. Corin Slown, an assistant professor in the College of Science. And she's been instrumental in this work and will be co-presenting with me today. So California State University, Monterey Bay, CSUMB, is pretty young, but we are very unique in the fact that we have UROC, an Undergraduate Research Opportunity Center, that has supported student success in a number of ways, including the McNair Scholars Program, a Research Rookies Program, and also a CURE Faculty Fellows Development Program that we'll be discussing in just a few minutes here. I work with Heather Haeger, Quentin Sedlacek, and Corin Slown on this program. And what we're really trying to do with this program is to cultivate a community of practice around cures. Okay, so in terms of scaffolding the cure experience and our goals at CSUMB, currently at CSUMB we have a number of upper division cures, including molecular genetics and bioinformatics. But these cures are often scattered in the curriculum, and like Frank mentioned earlier, in terms of goals for his campus, we really want students to experience cures throughout the curriculum. So we hypothesize that by scaffolding cures throughout the curriculum, students will experience at least 3 cures and will gain competence in biology, analytical skills, and a sense of belonging in the sciences. When we think about scaffolding, we're really thinking about a linked experience for students in which faculty are encouraged to think about the before and after picture of their students. Our strategy is to think about where students are and what they need in terms of building towards those upper division cures, and we're specifically targeting lower division cures to improve the overall student experience. All right, so our CURE Faculty Fellows Program has 4 professional development workshops. Those are about 7 hours long, and is supported by the provost and also an HSI STEM grant. In those professional development retreats, we have learning outcomes, and we work on a number of topics, including equity and inclusion, SMART learning outcomes. So are your learning outcomes specific, measurable, attainable, results-focused, and also time-focused? And then more recently, we thought about complex instruction and thinking about what our students are doing in our cures in terms of collaboration. We compensate our CURE fellows for their retreat time and also their 40 hours of course development time. And in order to cultivate a community of practice, we have our faculty fellows paired in mentoring relationships with folks that have already established cures on the CSUMB campus. In terms of assessment, we've developed a cure survey that out of the IRSA and LCAS that measures indirect student evidence. As seen by the light and dark blue bars on the bottom of this slide, students do not identify as scientists more in upper division courses. with light bars being students that don't identify as scientists and dark bars being students that identify as scientists. These data were collected at the beginning of the courses listed in response to the following question. Do you identify as a scientist? Why or why not? And we were really intrigued by the fact that there were no difference between our upper division and lower division courses. And so we decided to dig in deeper by doing focus groups. And today I'll focus on the data from one of our cures, that is a microbiology class, Biology 320. And this is really based on the student availability and, of course, willingness to participate in these focus groups. We were quite encouraged by the themes that emerged in the focus groups in terms of accessibility to cures and authentic research. For example, many of the students mentioned that the class was their opportunity to participate in research. We even had a transfer student talk about the fact that they lacked connections and resources needed to participate in research on campus, and that the microbiology class was their opportunity to do so. We also had narratives about students discussing data, that results were different, weird, and confusing, and that there were unknown outcomes. We were also really excited to see that the students matched in terms of what was collected for our cure survey, that's over on the left-hand side, sorry, on the right-hand side, and what was happening in the focus groups. So as seen on the far left, the dark blue bars represent a great gain in analytical skills, and this is student-reported data. And what we see here is that in microbiology, which is on the top, students are reporting great gains in analytical skills. We were even more encouraged to see that when comparing General Biology 1 and General Biology 2, so the bottom two bars there, that there's an increase in analytical skills reported there as well. This upward trend really matches their trajectory towards graduation and aligns with our hypothesis about analytical skills being increased with scaffolded cure experiences. With that, I'll hand it over to my colleague, Dr. Corin Slown, who will talk about direct evidence of student learning.
So one of the things that we want faculty to think about as they're planning and designing their cure is the way that their learning outcomes for the course map onto both the major learning outcomes and then ultimately our university learning outcomes for students as well. And so one of the things we ask faculty to do is to think about what are direct evidence measures that they could do in their class that think about components like content knowledge, but also think about skills and insights such as quantitative reasoning or critical thinking that would be broadly transferable to their future careers. And so here we have an example of work by a faculty member in the general biology course that Corinne White referenced earlier. And so there are several pieces of data that this faculty member used to triangulate and think about changes both in the course prior to the cure experience and then in 2 iterations or 2 semesters worth of that course with the cure components included. And so the first set that you see there are data looking at a concept inventory. And then the next set is data actually looking at students' ability to use quantitative reasoning to solve an exponential growth problem. And then the set that we don't have pre-data for is specifically the application of that exponential growth to the context of the problem with the cure. And then the last 2 sets are really thinking about their understanding of restoration. So this is the cure in context. So similar to what Amelia spoke about, this is a cure that actually has a field experience component in a regional, local context that looks at restoration and ecology specifically. And then the last set of bars is really thinking about students' framework of knowledge and how they connect that knowledge within a concept map. And so we actually see across the student experience this increase in gains. And again, when we're thinking about this, the error bars are 1 standard deviation. And so we're actually able to see some significant gain specifically in the second iteration of the course where the faculty member took the data from their indirect and direct evidence and made adjustments to their curriculum and to their pedagogy. So we're encouraged to see growth in our faculty fellows based on the assessment design. when we think about the CURE Fellows experience, part of what makes this a rich experience for faculty is the participation across multiple disciplines. So as Chris Myers alluded to, one of the things we're really trying to think about is not just science in practice, but scholarship in practice broadly across our community at CSUMB. And so while the first iteration of the CURE Fellows, the first year in which we did this faculty development, was almost exclusively STEM. We had one brave faculty member from our psychology department who was really thinking deeply about the way she wanted to integrate this into their research methods curriculum. We did have representation across biology and chemistry, primarily within our College of Science. But in the second iteration of our CURE Fellows faculty development program, one of the things we were encouraged to see was the participation from across our campus. And so we, in addition to having participation from computer science, we also had participation from our visual and performing arts, from business, and from our Japanese program, or the work that that course is doing within the context of history and food. And so we wanted to show this because part of what we think enriches the community of practice is really the diversity of perspective in terms of scholarship in practice across disciplines. So when we think specifically about scaffolding within then a major or a discipline, we're really thinking about strategically positioning faculty in the courses that they teach across the curriculum. And so you can see here an example of the biology curriculum, and this is supported through a Keck grant as well as the HSI STEM grant to really think about the courses that are in blue that faculty have already developed, and then the courses that are in red where faculty are currently in development. And so our thinking here is that we really want to think across the 100 to 400 course level about where students experience cures, and then is there coherence in that cure, and so can they think about how they would transfer skills, say for example, their analytical skills from the first semester of general biology to the second semester of general biology, and then ultimately to, say for example, a microbiology course. And so we have data to support that students do in fact transfer those skills, and that their ability to analyze data increases across that scaffolded experience. But we also recognize that especially as students move up into upper division courses, one of the things that ends up happening is they start to differentiate in their pathways. They're starting to identify their career aspirations and what is the most logical path for them. And so that's the point at which we think that there's also an opportunity to differentiate and really think about what skill sets are most necessary for an emerging workforce and what will allow our students to be competitive when they step into that market for the careers that they desire. So as we're thinking about the third iteration for our CURE Faculty Fellows program, there are a couple of pieces that we're really thinking about. Part of our thinking is how do you sustain this and scale this both within departments but then across colleges on our campus. And so some of the work actually that Quentin Sedlacek is doing is looking at the focus group data and really being able to share out with faculty the learnings and the insights from students that we think, as student voices drive practice, will encourage other faculty who perhaps haven't considered cures to potentially think about incorporating them into their practice. And that would be scholarship broadly, not just scholarship within the College of Science. So in 2019, we have over 900 students that have participated in a cure so far. And what's really exciting about this is that this semester represents the first time where we've had students have up to 3 cures potentially. And so one of the ways that we're excited about measuring the data is thinking longitudinally. This next semester will be the first time that we'll have students who are graduating, and so we can start to look at our graduation data around student participation in cures and ultimately their employment, so where they end up in their career or whether or not they're applying to graduate school. And so those are the other components of the data that we're really thinking deeply about analyzing. And so when we think about this longitudinal data then for multiple cures, there are a couple of ways that we're intrigued about how we're going to track and make sense of this data. Part of that is following students across an academic pathway, but that's also something that we want to think about in light of retention and persistence, specifically addressing the question of achievement gaps for our underrepresented minority students. And so with that, we're really excited about the next phase, And it's my pleasure to introduce our colleagues in the math department. So Lipika, Deka, Peri, Shereen, and Jeffrey Wan are actually going to be able to share out about the work that they've done in mathematics with respect to cures.
Hello, everyone. I'm Jeffrey Wand.
And I'm Peri Shereen.
I'm Lipika Deka.
And we're going to be talking about building cures in a mathematics major pathway. So our journey with cures really started with obstacles that I'm sure a lot of you face with your own research, but we sort of struggle with in mathematics a lot. Firstly, it takes us a long time to get students ready for research. For our research area in particular, we spent years in graduate school just to, get caught up to speed, and so it's a struggle to get students ready for our research. And two other obstacles which I think are related. One, there's a lack of motivation for students to do math research, and I think that stems from a lack of knowledge of, what is math research. A lot of time we get that students think we do calculus problems, but that's like 450 years ago. We've moved on since then. So we decided to become Cure Fellows in the summer of 2018 to sort of overcome these challenges and see how we can better introduce research to our students. But once we started, we kind of hit a roadblock. Math cures, well, at least from what we could see, are not really fully developed in the literature. There is some stuff out there, but we couldn't find a lot. So we decided to, define what is a math cure. What do we want our students to get out of it? So the first two objectives of our cures are related to what we do as mathematicians in research. So first, we want students to participate in mathematical research by generating research questions, developing conjectures, proving or disproving their conjectures, and presenting the results. And how do they come up with these conjectures? We want students to make discoveries unique to themselves in content that is not part of the current curriculum. So calculating counterexamples, searching for patterns, and making meanings of their examples. And I do want to point out, number two here, the discoveries that we're having students make during our cures are not necessarily unique to mathematics. Again, it's hard to get students up to speed and sort of at the forefront of research. So our cures are really meant to be making discoveries that are unique to them, and it's part of something that's, it's not part of the current curriculum. They're not going to see it in another class. And lastly, we thought it was really important that students develop their own mathematical identity. So our third objectives sort of hits on that. So we want students to develop their own math identity within the math community by exploring topics mathematicians are currently researching and researching within and with the community of their peers and doing some kind of summative research project. So this is the pathway that we generated, or we're coming up with. So if you see the four bubbles that are sort of stacked on top of each other, these are the classes that we've planned or have implemented a cure in. And the idea is that a student will take a cure in each one of these classes, and by the time they're out, they should be ready to do some kind of research problem in our area of Lie theory or representation theory or combinatorics. The first two classes, or at least the first row of classes, that's sort of the entry-level classes our math students take, and then the bottom two are the more advanced classes. Now, the classes that you see in green, these are the classes that we have made cures for, and today I'm going to talk to you about the cure that I did in differential equations / linear algebra. So my cure was a semester-long project that they worked outside on class on with 5 exercises and checkpoints that mirrored the content from the course. I'll talk about that in a little bit. The first time I iterated this, I made the cure optional because I was a little hesitant. I thought maybe it'd be too much for students, but after that first semester, I was like, no, everyone can do this. And so the second time I iterated it, it was mandatory. Students who participated got into teams of 3 to 4. I required that they met with me weekly, and these were student-led meetings, just to get, up to speed on what they're doing and to talk through if they're having any issues or any problems and just sort of what they're discovering as they're doing their exercises. Each team was allowed to submit 1 draft of each exercise for feedback, and then all drafts in the final product were typed using LaTeX, which is what we use in math research. So this is the general flow of my cure. we would do something in class, so class topic 1, and then students would do an exercise outside of the class that was part of the cure. So for example, we would learn about vector spaces in class, and then students would explore Lie algebras, which is just an extension of vector spaces. But it's an extension, though, that they're not going to see in a current, or in their current curriculum, and they probably wouldn't see it in a research project, at least over here. So this is roughly what would happen, though. We would do something in class, they would do a cure topic. We would do something in class, cure topic. And again, these cure topics were things that were extensions from the material of the course. Now, like Corin said, after we do a cure, we do survey our students. And I know she said this really fancy. We used, the surveys were created using constructions from URSA and LCAS. We did have to adopt, or we had to modify them so that they have more of a mathematical context to them. But in any case, I want to present some data that we obtained from this. Now, you're going to see a couple of slides that look like this throughout the presentation, so let me give you sort of a brief intro to what's going on here, because there's a lot to it. So if you look at the top middle, that's the theme of this slide, so motivation, one of our obstacles that we're facing with math research. And so we had students reflect on motivation by sort of looking at these 5, if you look at the middle right, these 5 statements here, and they reflect on these and they answer the question in red compared to before doing your most recent research, so cure, how likely are you now to agree with the following statement? And so the data we obtained is the middle left there. So if you see that bar graph, blue is good, brown is, we need to improve. So the 2 things I wanted to point out to you are the top and the bottom, or at least the first and the last question there. So learning mathematics is interesting. 58% of students agree with this after doing our cure, which I think is really good because that's what we want. We want students to be more interested in what we're doing and in math research. And then one thing that was kind of surprising to me was the last statement there. Learning mathematics will get me a good job. 64% of our students felt that after our cure, yeah, they do agree with that, which I thought was really interesting. Okay, I will pass it on to Peri Shereen.
Okay, well, I'm going to talk to you about my implementation of a cure into our Foundations of Modern Math course. This is our Introduction to Proof course for our math majors. It's a gateway course to the upper division courses that they'll be taking. It has 2 prerequisite courses, Discrete Math, which Lipika will talk to you about in a moment in her implementation, as well as Calculus 2. The class size is around 15 to 25 students normally. In my fall 2018 implementation, I had 24 students. I did implement it again this past semester with about 14 students. And a good majority of our students are upper division transfer students. And then one other thing I should add about this course was there was an existing class portfolio before I implemented the cure. And what that was essentially a proof portfolio that focused more on the writing of mathematics. And so what I did was I implemented the cure by expanding the portfolio to include 2 pieces, one being the writing portion that already existed, and the other being what I called the discovery, but really the cure piece to their sort of semester-long class project. So basically just quick overview of how it worked. The big idea is I wanted them to learn how to use the OEIS, which is the Online Encyclopedia of Integer Sequences, and is a very useful tool that math researchers use in combinatorics. And so what they did essentially was they asked a mathematical question that was the same across the groups, and I should say they worked in groups of 3 to 4. And then they, in answering that question, started to go through the process of developing conjectures and looking for patterns. And then they were able to use the OEIS to either confirm the patterns they were seeing or to disprove the patterns they were seeing. And then from there, the encyclopedia gives a lot of information about what's known around this. And so they started to ask and make connections to other mathematical areas. And then from there, they developed a research question around the connections they were making to this first part of the project. And then the final piece of it was writing a poster presentation that they submitted with their portfolio at the end of the semester. And as Jeff said, too, a good part of this was meeting with me several times throughout the semester to check in, to explain what they've been doing, to explain where they're stuck and where their hurdles were, and to sort of help mentor them through their project. So that's there's the big overview of what that was. And then I was going to present some more of our data for you. So this slide is about self-efficacy. And again, similar setup about how much gain they felt they had in the 5 questions to the right there. And again, more blue is what we're looking for. And so just something we'd like to highlight is that we saw that students felt they had progress on comfort, on working with others, and the ability to work independently, which is a big deal for us. The next one we looked at was discovery and relevance. And then the question here being, during this most recent research project, I was expected to, and there's those 5 questions. Again, more blue is what we're looking for. And so here we see that students felt they had progress on conducting investigations to find something new to themselves, which Jeff was speaking to earlier, right? For us, it was about them finding something new to themselves and other students and the instructor. And then lastly, we looked at collaboration in the classroom and tried to see where students felt along the 5 questions listed there. And so highlighting that we saw that students felt encouraged to discuss their investigations with their peers and instructors, as well as reflect on their learning. So these are some outcomes that we saw as a result of our first implementation of our cures. And from here, I'm going to pass it off to Lipika to talk about discrete math.
If you remember our pathway in the beginning, this is actually the first class where our students will see cure first if they come through our pathway as a freshman. This class is also designed not only for mathematics major, this is also a requirement for computer science major. And this is a class where typically they would get introduction to the formal language of mathematics, about argumentation, and they also learn several discrete structures. So this, in this course, the cure is kind of designed like a 322-hour proof class, but this runs throughout the semester. Also, the difference is this is a course where we have multiple sections, so it's a much larger implementation. So here, again, student works in groups of 2 and 3, 2 or 3 typically depending on our class size, and we will typically have about 9 or 10 groups in each section, about 3 sections a semester. So the CURE project, we aligned it kind of parallel to the course, and they would get support outside of class to work on it. And it's the whole project is divided into 3 different parts, and I'll tell you how each of the parts works. We, on the first part, we focus on helping them student to learn how to formulate a research question. So here they were given a new word or few words to investigate, figure out what relevant body of research exists for those words, and then learn through that process of how they can formulate their own research questions about them. They summarize their finding here as a group paper. And then in the second part, they actually learn, they focus on one of the questions they came up with in part 1, and then they try to investigate that more by looking at examples, counterexamples, working through various processes. Some students actually also create computer programs here to generate their examples, and having the benefit of computer science major in these groups makes it a lot easier to do that. And they try to see if there is a pattern and if they could conjecture an answer to their original question. And the last part is where they actually try to prove or disprove their conjecture, either by giving a formal proof or showing counterexample, or if they couldn't come up with either of them, at least talk about their challenges, because that's what sometimes expected in research. So this whole core project was done outside of class, so we try to support them as much as we can, like our other cures, providing them as much one-to-one time. And then we used online office hours, emails, Google Docs to try to communicate, because we had a lot more groups here, about 9 or 10 groups each. So we had to be a little bit innovative, but we made sure that they had enough support to get through and stay engaged. So wanted to show you guys what we learned along this process of implementing our cures. I think several of these came out from previous presentations, so we're kind of seeing similar trends, but we learned that we need to be a little bit flexible about how each group works and their process. We also learned that sometimes our final product for a particular group might not be exactly what we are expecting, but this is research. And then we realized one of the things, particularly for Math 170, that having one-to-one meeting, which has amazing positive output, but was very time demanding for the instructor. So we're trying to think how we can do that better or easier. With that, I will go back to sharing one more slide on data. This is, again, very similar to what you saw before, but the focus here is on analytical skills and how students felt, how they are doing through their experience of cure. And then again, more blue means good, and we did see some positive results here, which is encouraging, and wanted to highlight that students felt, which is important, that they are making progress on their problem-solving skills, and particularly figuring out what is the next step in the process. We also wanted to finish our discussion with a slide where we compare the first implementation of Math 170 and second implementation, because we are able to finish 2 and we have actually into our third. And I know I have a lot of data here, but what I want you to focus on is the blues, because remember, more blues means good. And the first column here shows you the blues on the first and the second on the second column. And it's really encouraging for us to see that even within one implementation difference, we see more blues, and that's very positive, and we hope that it stays that way. With that, I want to thank our organizer for organizing this webcast, and also want to hand over to Frank for discussions and questions. Thank you.
Thank you, Lipika, Peri, and Jeffrey. All these projects are quite impressive, and I'm certainly excited about all the great work and how it's impacted our students and future leaders of tomorrow. We have a little bit of time for Q&A. I certainly thank the registrants for providing their questions online. Here's a question. I think this would be appropriate for Chris. What are some of the costs associated with the different kinds of cures, and what resources are you using to support and sustain cures on your campus?
Well, that's a great question, and I'll let the audience know if you promise not to tell anyone, and that is cures are more expensive than if you have a shrunk-wrapped lab. But I think the gains, as you see from the assessment that's been presented, are fantastic, and we can't not do this. So what are some of the extra costs? Supplies, because again, if you're collecting research quality data, particularly in molecular biology, that can get expensive very quickly, and most of us don't have really big budgets for lab supplies. There's a cost in personnel or human resources. One thing that's often forgot is the role of staff, and that's because cures tend to be evolving and changing, and you have to be flexible, as has been mentioned. That is a little more time intensive for staff. It also requires more supervision in the classroom because not everyone's doing the same boring thing that they're Googling to look up what the answer is. And so in an ecosystem in a course, you saw from the Journal of Chemical Education that the idea of a postdoc or a grad student collaborating with the professor, near peer mentors that perhaps have been through the experience, and so that's a little different than the traditional TA. So these are extra costs. So okay, part 2 was where is our money coming from that? And so I really recommend a diverse portfolio of funding. That would include grants. Obviously, National Science Foundation has come up several times. They're big supporters of cures, as well as HHMI. But also, we've found a lot of success with donors and philanthropy, in that we, in interacting with a donor that ended up giving us $500,000, first one thought was why can't we do this in maybe student scholarships, and then I kind of counter-propose it. That's kind of one student at a time, and there's a lot of scholarship money for students, and sometimes that cuts into their financial aid, so it's not. What about supporting the infrastructure of a program like Cures? And they really bought into it, and so to have an endowment that can pay for it, that's a very powerful thing. I think this is a very good use, and I'm sure everyone will agree, a very good use of CSU student success money. Rather than you're having a direct impact on that graduation rate. Intramural funding to help develop cures. As I mentioned, one of Tricia Van Laar's project was funded by CSUMB. And then if you have science education folks in your area, that kind of ties into their research area. Yet we're fortunate, Emily Walter in biology, Dermot Donnelly in chemistry, just to name a few, engaging those with other research support to add up to support this. So so those are just some thoughts.
Great, thank you, Chris. And hopefully your affluent donor has some affluent friends that can provide even more money to the CSU.
Well, that's what I'm working on every day. Yeah.
Well, hopefully you can spread the wealth there.
Yeah.
We have another question here, and I think the Corins, I know there's Corin and Corin. What if there are multiple instructors for a course with a cure?
So we actually have several faculty that have wrangled that question, and we found that actually engaging in a community of practice can be really effective. So typically what has ended up happening is that there's been one faculty fellow who developed the cure, but then they work with a team of instructors that are teaching specifically the lower division courses. And so we, through UROC and then myself through the College of Science, endeavor to provide as much support as possible. So for example, when they're doing the indirect assessments, we provide support there, or Quentin Sedlacek, our awesome HSI STEM postdoc, supports with the focus groups. So it really is a team effort. And I think more than anything, we're trying to make sure that there's coherence across student experience. And so some of that is just communication on our part. But then it's actually really exciting too, because that larger amount of data, both in terms of the student experiences, but also the instructor insights, actually helps refine the next iteration when teaching the class. So I know that math has seen that in terms of conversations with their instructors. Biology has seen that, where the feedback from multiple instructors actually helps to refine the course and the cure experience.
David, here's one specific for you. You mentioned biomaps as a potential good assessment tool for cures. Can you comment a little bit more as to why this it might possibly be a good tool for assessment?
Yeah, so biomaps is a validated content knowledge assessment tool. Just like many of the others, it has a broad range of questions that span all of biology. But the really cool thing about biomaps, as I understand it and trying to implement it, is that you can actually choose the questions, specific questions out of the question data bank to evaluate. So you could choose questions that are more relevant, more directly relevant to the actual content covered in the CURE course. Many of the ones we tried to use in the past are too broad, and you can't really tease away those questions, and so it doesn't really give a good assessment of exactly what was covered in the course.
Okay, thank you. And I think we have time for one last question for the math people here. How are these cures different from the science cures?
I would say probably one major difference, I mean, at least from my understanding, is that our cures are not novel to the mathematical community. We're usually having them explore things that have been already well developed in a sense, but our projects are all things that they won't usually see in an undergraduate curriculum and are not part of the standard course. And at least in my class, they're graduate level topics. So it's not new to the math community, but at least it's new to them. And I don't know if any of the other math people want to add to that, but that's one difference.
Yeah, I would agree to that. I mean, I think that's the challenge for math research is being able to get them in to doing research quickly. And so for us is about the experience and what they're doing throughout the cure, and that it's novel to them is the idea.
Okay, thank you. I'd like to thank all of our speakers today for presenting on their Cures projects. They're very wonderful, and I applaud the great work that our faculty are doing to broaden research opportunities for our students. A link to the webcast will soon be made available for all the registrants today. I'd like to also thank Monica Alarcon, Administrative Analyst here at STEMnet, for helping a great deal here. I'd like to inform everybody that our next webcast will be at 10 a.m. on Wednesday, March 11th, focusing on CSU major institutional grants, focusing on both NSF and NIH. You can contact either myself or Monica at malacon@calstate.edu or myself at fgomez@calstate.edu for more information. Until next time, I hope you all have a great day. Goodbye.